We've already discussed how the sarcomere is the microscopic functional unit of skeletal muscle. And you can see that sarcomere here, the full sarcomere here, is going to run from Z line, Z disc, to Z line, Z disc. You're going to see that a couple of various zones are marked. These are the ones that you really kind of need to know. We already know about the Z line or Z disc, which are going to be the boundaries of each sarcomere. And the sarcomeres are going to be lined up just like railroad cars, dozens and dozens, potentially hundreds, if not thousands of them in a single muscle fiber, because remember, these are tiny, microscopic. We're also going to talk about these blue lines and these red lines. So the proteins that we need to know are going to be actin and myosin. What do you need to know about actin? Actin is thin. Actin is thin. It's going to look like two strands of pearls that are twisted together. Actin will also contain active sites. Actin will contain active sites, and hence the name actin. We also need to discuss myosin, which is going to be the thick filament. And myosin is uniquely configured. It is going to have these globular heads on it, and we'll see some images of these shortly, so hang tight. And those globular heads, to me, always look kind of like golf club heads. And those golf club heads will attach to the active sites when they are exposed. If you take a look at the diagram, you'll see that there's I band and A band and I band again, and then an M line, blah, blah, blah. If you take a look at the A band, here's the A band. The A band is going to stretch where the myosin is. It's going to be a little overlap with actin, but this is the end of myosin to the end of myosin. The I band is going to be just where the actin is, and the Z line is going to be in the middle of that. All right, let's push on. We've already talked about actin. Actin is thin. It contains the active sites. It is the one that looks like two strands of pearls that are twisted together. And we've discussed myosin. Myosin is the thick myofilament, and that is the one with the globular heads, the ones that kind of look like golf club heads. We also need to discuss troponin and tropomyosin, and I put these together because they form a complex which is conveniently known as the troponin tropomyosin complex. They form a complex known as the troponin tropomyosin complex. So let's take a look-see at what's going on here. This shows you actin. You can see that what look like two strands of beads twisted together, and you can also see the troponin tropomyosin complex. See how they're interconnected? The troponin is actually going to sit on an active site on actin. So here's an active site under this troponin tropomyosin complex. Here's another one, and there are going to be multiple more along every chain of actin. The myosin is going to have those globular heads, which are sort of confusingly known also as cross bridges. And it's confusing that they're known as cross bridges because they actually form cross bridges with the active site on actin. So when the head of the myosin attaches to the active site on actin, that's known as a cross bridge. That's a little confusing. Now, you'll notice that the active sites are usually closed by the troponin, blocked off by the troponin tropomyosin complex. And that is because your skeletal muscles are not always contracted. So let's sort of easily move into this. When the troponin tropomyosin complex moves off that active site, this globular head can actually bind with that active site. And that is the very beginning and basis of skeletal muscle contraction. So let's take another look-see. We know that we have a sarcolemma surrounding a skeletal muscle fiber, and we know that the cytoplasm of a skeletal muscle fiber is unique and called a sarcoplasm. We know that the sarcoplasmic reticulum is going to be quite extensive and unique to skeletal muscles. And we know that it'll also store calcium. This shows you a chunk of one of these myo fibers taken out, and you can see a sarcomere here. And this is a blown up sarcomere. So here, the yellow lines are going to be the actin, and the red lines with those golf club heads, those globular heads, will be the myosin. So I want you to sit back and not take notes on this, and you can go back and listen to what I say and take notes on it. I just want you, right now, I want you to sit back and listen and understand. Just put your pen down, or your pencil down. The active sites on actin are covered by that troponin tropomyosin complex until you contract your skeletal muscle. When you have a thought, I'm going to pick up a pen, or I'm going to scratch my nose, or I'm going to wiggle my toes, or I'm going to shift in my seat, that thought, whether it be conscious or unconscious, is going to trigger an action potential down a motor neuron. When that action potential gets to the end of a motor neuron, magic will happen, and information will go to the skeletal muscle fiber that you want to move. That will trigger a cascade of events, and yes, you can use that term. It is legitimate to use that term. That will trigger a cascade of events to occur, which will cause the release of calcium from the sarcoplasmic reticulum. When the calcium binds with the troponin tropomyosin complex, that will make that complex flip off an active site, and one of the globular heads of myosin can land there. That's what's known as a cross bridge. In order to break that cross bridge, you have to have an interaction with myosin and ATP. ATP hydrolyzes or breaks down to ADP. That connection will be broken, and the globular head will be ready to move to the next exposed active site. So in essence, what happens is this. I want you to imagine driving along Cass Park, and you look over to the inlet, and there are either the Ithaca College or the Cornell crew teams out there. And they're in those shells and or skulls, and they're rowing along. Imagine that those oars are actually the globular heads of myosin rowing from exposed active site to exposed active site to exposed active site. That means that the fibrils, those myofibrils, are going to slide past one another because myosin is ratcheting along actin. The Z lines will be pulled closer together. And that is a very, very, very high level explanation of what happens with skeletal muscle contraction. And we'll go over it again 6 ways from Sunday, so don't panic if you're not getting it. I put this here because we're going to go over what I just said before. In the top diagram, the sort of, I don't know, peachish, orangish structure or lines are going to represent myosin, the thick filament with the globular heads. The green lines show mostly actin, although they also are the color of the Z lines. And remember that the sarcomere runs from Z line to Z line. The bottom part of this diagram is great because it shows you actin with the active sites covered by the troponin and tropomyosin complex. So let's expand what I said before just a tiny little bit. you have a thought, I want to move. That information is translated from a part of your brain down the spinal cord out to what's known as a motor neuron. Motor neurons only carry motor information, and that information is always going from the central nervous system out to the periphery, always, 100% of the time. That information gets carried over to the skeletal muscle fiber, and a cascade of events will trigger the release of calcium from the sarcoplasmic reticulum. So why do we care about calcium? Because calcium is what needs to bind with the troponin tropomyosin complex in order to have it roll off the active site. Once that active site is exposed, the globular head of the myosin can attach to that active site and will stay there until the ATP interacts with the globular head. It gets hydrolyzed. You have ADP in a phosphate group. It'll break that cross bridge or interconnection, and the globular head will be ready to move to the next exposed active site. What do you need in order to have a skeletal muscle contraction happen? Write this down, and you need to know it. What do you need to have a skeletal muscle contraction? You need an action potential, which is carrying the thought, the information from your brain down along the spinal cord, out to the motor neuron, and along the motor neuron. So the first thing you need is an action potential. You also need calcium. If you have insufficient calcium or no calcium, or the calcium can't be released from the sarcoplasmic reticulum, or isn't released from the sarcoplasmic reticulum, nothing will happen. No calcium, no skeletal muscle contraction. This shows you what we went over. On the left, you can see that the sarcomere is at rest. Remember that the Z lines are the boundaries of the sarcomere. The blue structures in this are representing, you got it, myosin. The red structures that are not Z lines are representing the actin. Actin contains the active sites. Those active sites will be covered by the troponin tropomyosin complex. Between the sarcomere at rest and sarcomere contraction and the filament sliding on the right, you have to have the release of calcium. Calcium will bind with the troponin tropomyosin complex. That complex will roll off the active site. A globular head of myosin will attach to that active site until an interaction with ATP that will cause the release of the globular head, and it will seek to move on to the next exposed active site. That will happen multiple times on multiple myosin and actin fibers, and that will pull the Z lines closer together, and that is a high-level overview of how skeletal muscle contracts.